Hello everyone, the project presented is being developed by a postdoc fellowship, Matheus Reis, coordinated in collaboration with Matheus Reis from Laval University and Nilva Sakomura from São Paulo State University. This presentation will highlight the most critical aspects of the project, modeling the growth of broilers and the metabolic fate of calcium and phosphorus. The ability to foresee the response of broilers due to nutrients intake might be crucial to the progress and sustainability of a poultry farm. Modeling the biological system is a viable approach to predicting broilers' response based on factors such as feed composition, environment, and others. In this context, a broiler growth model was developed in Brazil based on more than 20 years of research conducted at São Paulo State University. The model predicts the growth of the main chemical components of broilers and the requirements of energy and amino acids. However, it does not account for calcium and phosphorus. On the other hand, a research led by Dr. Matheus Reis at Laval University, in collaboration with INRA, developed a model to estimate the metabolic fate of calcium and phosphorus in broiler chickens. A collaboration between Laval University and UNESCO aimed to implement the calcium and phosphorus model in the broiler growth model, evaluate their outputs, and improve the predictions. Overall, the model can be comprehended as three interconnected mechanistic models. They are soft tissue and feathers model developed at São Paulo State University. This model is based on the broiler growth model and predicts modern broilers' daily feather-free body and feather weights. The main chemical components from feather-free body and feathers are predicted and added to estimate the whole body weight. The bone ash model was developed at Laval University in collaboration with INRA and University of Maryland. This model predicts the potential and the actual weight and deposition of calcium, phosphorus, and miscellaneous minerals on bone ash. Finally, there is the digestibility model, also developed at Laval University in collaboration with INRA. This model predicts the digestibility, absorption, and excretion of calcium, phosphorus, and miscellaneous minerals, considering many factors, such as the interaction between calcium, phosphorus, and phytase from feed and endogenous losses. The soft tissue model is predicted from the broiler growth model based on daily depositions of body protein and body lipid. Also, the feather protein and body water are predicted daily. The digestion model accounts for the flow of calcium and phosphorus from feed to the plasma, based on digestibility, endogenous losses, and excretion of calcium and phosphorus. Finally, bone ash model connects the model cited before, where we can highlight the following. Once calcium and phosphorus are in the plasma, they can be deposited on four distinct compartments: bone ash, bone protein ash, body lipid ash, and feather protein ash. The flow of calcium and phosphorus on soft tissue and feathers are priority over bone ash. Calcium and phosphorus can be deposited and mobilized from bone ash depending on the intake, absorption, and the necessity to fulfill the deposition of calcium, phosphorus on soft tissue and feathers. Body ash is predicted by adding the amount of calcium, phosphorus, and miscellaneous minerals found on bone ash, body protein ash, body lipid ash, and feather protein ash. To better understand how the whole model works, we need to look at each model closely. The soft tissue and feathers model can be summarized in the following steps, in which body protein is the primary driver of the model. The model starts with a prediction of feather-free body and feather protein using complex function. Secondly, water, lipid, and ash are predicted from protein using allometric equations. All the chemical components predicted are added together to reconstruct the animal. The first derivative of the complex function is used to predict protein deposition. Predicting protein weight and protein deposition is necessary because the requirements for maintenance and growth are based on body protein and deposition. To calculate the requirement for energy and essential amino acids, the necessity for maintenance and growth are added together. The amount of feed necessary to meet the requirement of the first limiting nutrient in the feed, being energy or essential amino acids, indicate the desired feed intake. In practical situations, however, it is expected that broilers are prevented from achieving the desired feed intake. Thus, in a tentative to mirror the biologic phenomenon, Factors such as the bulk capacity of intestinal tract, lipid deposition, feed composition, environment, and health status of the broiler were included in the model to estimate the constraints in the feed intake and the actual feed intake. From the actual feed intake, real growth and nutrient requirement are predicted in the output. The digestibility model accounts for the mechanisms that affect the absorption of calcium and phosphorus. First, the digestibility of calcium is calculated based on the concentration of calcium and phytase in the feed. Then, the digestibility of phosphorus accounts for the different forms of phosphorus present in the feed, non-phytic phosphorus and phytic phosphorus. In addition, the effect of calcium and phytase and their interaction with phosphorus are also included. All constants and equations were produced by Dr. Marie-Pierre's team at Laval University in collaboration with INRA. For simplicity, parameters will not be covered in this presentation. In the bone ash model, calcium weight and deposition potential in the bone is predicted using polynomial equations. The prediction of calcium weight and deposition in bone enables a prediction of phosphorus based on a constant of 2 to 1 between calcium and phosphorus in bone, and the prediction of miscellaneous minerals, considering the constant of 48% of miscellaneous minerals found in bone ash. These steps are straightforward, but they only account for the deposition potential. An integration among the three models applies, as demonstrated before, to account for the real deposition potential and the actual weight of each mineral of bone ash. As stated before, this is an ongoing project, and so far we have accomplished a combination of the three models mentioned, and they are now working on the broiler growth model basis. The mechanistic model is being evaluated basically in three distinct steps. A model behavior to test the major hypotheses included in the model, a global sensitivity analysis using Sobel indices, and an external evaluation approach to assess the accuracy of the predictions. Using global sensitivity analysis, the main parameters that affect the predictions of calcium and phosphorus deposition in soft tissue and bone ash were evaluated. Evaluating phosphorus deposition in soft tissue, global sensitivity analysis indicated that phosphorus concentration in body protein is critical to the estimation of phosphorus in soft tissue. Similar results are found for calcium. Evaluating the deposition of calcium and phosphorus in bone ash, the linear coefficient from the equation that predicts the deposition of potential of calcium has more effect. An external evaluation is being performed to evaluate the predictions of the model in contrast with observed data. For that, a database was constructed using published literature and personal data. The evaluation focused on the outputs from soft tissue, digestive, and bone ash models, as well as the excretion and retention of calcium and phosphorus in the body. When the evaluation is based on published literature, a mixed model is used to account for the study effect. The predictions obtained from various simulations are compared with observed data, and the decomposition of the errors is analyzed. A personal database containing observations from trials conducted in Brazil was used to evaluate the soft tissue and feathers model. Among the observed data, there were the three most used strains in Brazil, males and females. Broilers from this trial were fed with a standard feed in a thermoneutral zone to allow broilers to achieve their genetic potential. Data were collected every 7 days, and the response variables evaluated were body weight, body protein, body water, and body lipid. Partial results demonstrate that body weight has a lower overall error of 2%, and from that, approximately 90% is due to disturbances. What was followed closely by body water and body protein. The predictions on body lipid accumulate more error than other body components, accounting for approximately 39% of overall error. Nevertheless, the results indicate that more investigation should be done to understand this behavior. A database containing 41 published literature was used to evaluate the digestive model. The response variables evaluated were absorption and digestibility of phosphorus. The overall error on both response variables was about 11%, in which 99% of this variation was due to disturbances. 3 published literature were used to evaluate the bone ash model. The response variables evaluated were calcium, phosphorus, and body ash. The error decomposition indicates that around 23% of total error is in central tendency, which suggests a possibility of improvement in the model. A database containing 15 published literature were used to evaluate the dynamics of phosphorus on the body. The response variables evaluated were the excretion and retention of phosphorus. The error in central tendency observed for phosphorus retention is around 22%, suggesting that the predictions of this response variable could be improved. Significant references used to construct the model and the publications of parts of the model presented herein can be found in these references. We would like to thank the contributors of this project, Marie-Pierre from Laval University, Agnes Narcy from Minha, Ilva Sakumura and Matheus Reis from São Paulo State University, and Roselina Angel from the University of Maryland.
All right, thank you, Matheus. Nice work. So you've presented your progress on the model. What are the next steps? How long do you see it taking to complete the model and being able to use the model to I guess roll out the model?
Okay, great. So yes, now we are in the evaluation process. We hope that until the end of this year, we have the model inversed to calculate the requirement. And now?
Yes.
Oh, great. So we hope until the end of this year, we have everything completed, and we hope that we have the paper at least submitted to a journal.
Okay, great. So once the model is validated, are you going to be able to apply this model, obviously with some other factors, but to other types of poultry, whether it's turkeys or ducks or geese or laying hens?
Yeah, laying hens is very it's different. I don't think we can use the same model and apply it for laying hens. We have to add some other parameters and investigate this further. For turkeys and other growing animals, I think if we reparameterize the model, it might be suitable to be used for ducks and growing birds in general. But the model have to be reparameterized for the species.
Okay,